I think there are many problems to address. There's many interesting things. You look at astronomy, and all of a sudden there are questions that people didn't even know they were going to be asking 10 and 20 years ago, talking about dark matter, talking about dark energy, talking about how many planets are there. How are they going to be able to detect planets in the world? Our whole view of the sky has changed drastically in recent times, and it's wonderful. The same thing is happening in chemistry, the same thing is happening in physics, and very definitely the same thing is happening in biology. There's all sorts of areas now that we are starting to look at that we never had the imagination to think we could be asking questions about. We are really about to enter the time of detailed sequencing of everyone's DNA. Now, whether that will lead to individual treatments, I'm not sure. But I am fairly confident that it's going to tell us so much more about human biology when we really get a chance to look at the variation. And we're already researchers are being able to discover things about human biology from what's called deep sequencing. And that's going to change a lot of how we think about humans. We already now have, for example, this is maybe not the nicest example, but it's an example. We have something like 100 different types of bacteria in our mouths. Even if we brush our teeth, we have 100 different types of bacteria. And only, I think, about 3 can actually be grown in the laboratory. So we've not been able to study them at all. But the sequencing of all of their DNA has now opened up their biology to us as well, and to allow people to start thinking about what these other organisms that are integral parts of ourselves. We have more cells in us, in our mouths, in our intestines. We have more cells that are bacteria than are human. And they're all part of this organism that walks around and talks and everything. And people are finding that these bacteria in the intestine have profound effects about the development of the intestine. They have profound effects on when we take a drug. Some bacteria cause the side effects that the drugs have, the unwanted side effects that the drugs have, because they're changing the chemical. So all of a sudden now people are starting to say, we really need to understand much more about these bacteria. So bacteria were the organism to study in the 1950s and 1960s, and then people gradually in the 70s and 80s and 90s. And now it's been more looking at animal cells. All of a sudden, with these new techniques, bacteria are suddenly fascinating. And we're learning an enormous amount more, including that bacteria are not single-cell organisms that just go off on their own and don't do anything. They communicate with each other. And looking at how these bacteria communicate and interact is another way that we're learning, in some cases, even about how human diseases are caused by these bacteria, because they cause the disease when they are in an aggregated state, not when they're isolated. So a lot of fascinating things are happening. That's all opening up. When people finished the sequencing of the human genome, sometimes it's said it was 10 years ago, it was not. It was fewer years than that. When we finally had the real sequence for the human genome. And when people could estimate what genes there were. And of all the genes that are there, we can look at the sequence and we can predict what protein is made. But for the vast majority of those proteins, their label is protein of unknown function. We don't know what it does. So although we have the sequence of the DNA, there is most of what the genome makes is an unknown to us. And it goes beyond what each individual thing works. It's also, how do all of these interact? How are they regulated? Because all of these are in a complex, and people are starting to study this now. So it's a very exciting time to understand a lot about biology in general and human health as well. There's lots to learn. As people develop, usually is in science that a technique will be developed, and all of a sudden people will see this is wonderful, and sometimes they oversell it by saying this will answer the question. More likely, it's this will lead us to new questions, and we won't know where we're going, but we will learn something at a greater depth than we had before. Whether it answers the question we're hoping it will is not clear. I want to say one other thing about this. I don't want to give the impression that just studying this in people is going to be the only way we're going to learn. There were some people, there still are some people in the United States that have the feeling that science has learned so much, that's correct, that we don't have to learn anymore. That we can just apply what we've learned to human disease and human welfare. And we're done. We've done it already. We've learned so much because we have learned so much. There's been an explosion in important information. But it's not that we have the answer. And we find, for example, in this tiny little worm that I study, people over the years have discovered completely new types of genes that then have been found in mammals, including humans, and other organisms that people didn't even imagine were there. But by studying in one organism, you get a chance to see it if it works in another. You use different systems to address problems, and as you make discoveries, you see if it's generally applicable. So there's a lot more that we have to understand. I want people to know about me is what the experiments were, what I've discovered, what I think is the implications of the work that I'm doing, and hopefully have an appreciation that that work has been useful. It's not, as I said on the stage, every scientist has to justify what they do. That they justify to get a grant. But once we've justified it, then we try to answer those questions. But if a new question comes up, if a new direction is there, then that's a wonderful thing to have happen. So you want to have that. And I want to be known for what's come out of my lab, even the mistakes that have come out of my lab. But I want to be known for the results, not for my personal life. It doesn't make any difference if I play the guitar or I don't play the guitar, right? It's irrelevant for the science. And the same thing is for beliefs. I'm not very organized. I mean, I have a somewhat neat office, but I'm not, the work is more disjoint. Creativity is, it's a funny thing, but an astonishing number of scientists are also musicians. That they have a real joy in that. I try pitifully badly, but I try to compose on the guitar. And it takes a long time. I get an extreme amount of joy from it, stumbling through and doing what I can do on it. My feeling is that a lot of science is creativity. If you just blindly go and do things, no one blindly goes and does an experiment and then says, okay, I've done the experiment, I'm finished, now I'm going to do another experiment. Every paper we write has an introduction, a results session, and a discussion, thinking about what the implications are of the work that we do. And while we're doing the work, as information comes up, we say, what does this mean? And we're always trying to make connections, or at least many of the people I know are trying to do that. But it's a funny sort of thing because you've got to keep doing the work. You have to have the information in order to be creative. You can't just sit there and say, oh, I'm just going to be creative. There are some people that can do it. I'm not. I'm a person that needs to have results that I can think about, that are sort of the basis. So I think you need both. You need to be able to do the work. If you don't have the work, you can't have something to think about. There's a wonderful phrase that I learned a couple of years ago by the famous hockey player Wayne Gretzky, who said, "I miss 100% of the shots I don't take." In other words, you got to keep trying and doing different things and seeing what comes out of it, and then working with what you have.